Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Streptococcus agalactiae, often known as Strep agalactiae, is derived from the Greek words strepto, chain, coccus, round, and agalactiae, no milk. So, Strep agalactiae is the spherical bacteria that grows in chains and was previously known to infect cattle, resulting in decreased milk output. Later, Strep agalactiae was shown to be a human potential pathogen, causing a variety of illnesses that mostly affect pregnant women and babies. Strep agalactiae is also known as Group B Strep, GBS, in the Lancefield classification, which was devised by American scientist Rebecca Lancefield. Now, a brief overview of microbial anatomy and physiology. Strep agalactiae has a thick peptidoglycan cell wall that absorbs purple dye when gram-stained, indicating that this is a gram-positive bacterium. It is non-motile and does not produce spores. Additionally, it is a facultative anaerobe, which means it can thrive in both aerobic and anaerobic settings. Streptococcus species are catalase negative, which means they do not generate the enzyme catalase. This differs from other common gram-positive cocci, such as Staphylococcus, which are catalase positive. When grown on blood agar, Strep agalactiae colonies produce beta-hemolysis, commonly known as complete hemolysis. That's because Strep agalactiae produces a toxin called beta-hemolysin, which induces total lysis of hemoglobin in red blood cells, causing blood agar to turn from red to translucent yellow surrounding the colonies. However, some Streptococcus species, such as Strep pyogenes, are beta-hemolytic. To particularly identify Strep agalactiae, use the bacitracin, hippurate, or CAMP tests. The bacitracin test involves adding a disc of bacitracin to the blood agar. Strep agalactiae is bacitracin resistant, so the colonies survive. Strep pyogenes is bacitracin sensitive, so the colonies die. In the CAMP test, Strep agalactiae and Staphylococcus aureus are cultivated on the same blood agar. Both of these bacteria are beta-hemolytic, but Strep agalactiae produces a chemical called CAMP factor, which promotes the activity of staphylococcal beta-hemolysin. This causes more beta-hemolysis when the colonies cross on the blood agar plate. Finally, the hippurate test relies on the fact that Strep agalactiae is the only strep that makes hippuricase, also known as hippurate hydrolase. To determine this, a colony of bacteria is transferred from the culture to a test tube containing a specific hippurate media and incubated for 2 hours. Then a few drops of ninhydrin are added to the mixture. Hippuricase, when combined with Strep agalactiae, transforms hippurate to glycine and benzoic acid, and glycine interacts with ninhydrin, causing the mixture to become deep blue or positive. Okay, Strep agalactiae possesses a multitude of virulence factors, which act as assault weapons, allowing it to attack and destroy host cells while evading the immune system. First, Strep agalactiae is encapsulated, which means that it is surrounded by a polysaccharide layer known as a capsule. The capsule also contains pili, which are hair-like extensions that aid bacterium attachment to a host cell. In addition, the capsule contains sialic acid, a chemical present in human cells. Inexperienced immune cells, such as those of a newborn, may mistake Strep agalactiae for self cells, allowing it to live within the body. Finally, Strep agalactiae produces beta-hemolysin, which damages host tissues and induces hemolysis, which means it eliminates red blood cells. Surprisingly, Strep agalactiae is so widespread that it may colonize up to 30% of healthy persons without producing illness. They mostly colonize the gastrointestinal system and vagina. Problems develop in those with compromised immune systems, such as infants, pregnant women, and adults with underlying conditions such as diabetes, mellitus, cancer, or a previous infection such as HIV. Furthermore, because the spleen plays a key role in immunization against encapsulated bacteria, Strep agalactiae infections are more likely in persons who have had a splenectomy, which means their spleen was surgically removed, or in sickle cell disease patients who have functional asplenia. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Strep agalactiae can cause many infections. Bacteria can enter the uterus through the cervix in pregnant women colonized with Strep agalactiae. From here, they can infect the fetal membranes, specifically the chorion and amnion, producing chorioamnionitis. When this occurs, the membranes might become damaged and rupture before birth, resulting in a miscarriage or preterm delivery. Sometimes the illness might spread to the fetus, resulting in intrauterine mortality. Alternatively, Strep agalactiae can enter the vagina via the perineum and travel up the urethra, producing cystitis, which is inflammation of the urinary bladder. Pregnant females colonized with Strep agalactiae are also at risk of passing the germs to their infant during natural delivery. Strep agalactiae is more likely to enter neonates' airways and proceed via the respiratory tracts into the lungs. This can result in inflammation of the lung tissue or pneumonia. Strep agalactiae can occasionally damage the baby's alveolar lining with their beta-hemolysin, breaking through the alveoli capillary membrane and entering the circulation, causing bacteremia. Bacteremia can induce newborn sepsis, which occurs when germs create a broad immunological response, resulting in vasodilation, hypotension, and hypoperfusion of the baby's essential organs. Alternatively, bacteria from the blood can pass the blood-brain barrier and enter the CSF, resulting in newborn meningitis. Less common, but still possible, the bacteria might migrate down the bloodstream to the joints, resulting in septic arthritis. Antibiotics such as penicillin G and ampicillin are required to treat Strep agalactiae. Cefazolin or vancomycin can be used to treat penicillin-resistant strains or those who are allergic to penicillin. There is presently no vaccination for Strep agalactiae. Hence, the only option to avoid infection in newborns is to provide colonized moms intrapartum antibiotic prophylaxis, which means giving antibiotics a few hours before birth. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. To summarize, Strep agalactiae is a gram-positive spherical bacterium that typically lives in chains. It is beta-hemolytic, non-motile, non-spore-producing, catalase-negative, hippurate-positive, bacitracin-resistant, and CAMP-positive. This bacteria has the ability to colonize the gastrointestinal system and vagina without causing symptoms. Most commonly, it causes newborn diseases such as pneumonia, sepsis, meningitis, and septic arthritis. However, it can also induce chorioamnionitis or cystitis in pregnant women. Penicillin G or ampicillin are the primary treatments, while cefazolin and vancomycin may be used as alternatives. To avoid Strep agalactiae infections in newborns, vaginally colonized pregnant females can receive intrapartum antibiotic prophylaxis. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you.